Atomic Force Microscopy, or AFM, uses mechanical probing of a surface to generate high-resolution images without the need to chemically treat the sample. In this video, bacteria are immobilized onto gelatin-coated mica and imaged in a liquid environment by AFM. First, mica squares are cut to fit the AFM microscope platform. The top layer of the mica is removed and the mica squares are coated with gelatin. Next, a droplet of bacterial suspension is placed on the gelatin-coated mica and spread using a pipette tip. The samples are incubated for 10 minutes, then the unbound bacteria are washed from the surface. The sample is placed in the AFM wet cell and imaged to obtain high-resolution images of live bacteria.
The main advantage of this technique over existing methods like isoporous filter immobilization is that both rod-shaped and spherical bacteria can be reliably immobilized. Also, a greater percentage of the cell surface is accessible to the AFM probe.
A key advantage of atomic force microscopy over other microscopies is that single-cell studies can be conducted. Investigations into cell morphology, septum formation, and cytokinesis can be pursued.
Individuals new to this method will struggle because they tend to use gelatin that's not proven to immobilize bacteria, or they use bacteria grown in a complex media where components will compete with the adhesion of the bacteria to the gelatin surface.
We first had the idea for this method from experiments back in the 1970s, where bacteria were immobilized on gelatin.